Okay, so now we're going to be using some whiteboard to actually look more at our eukaryotic cell. And the first thing we're going to do is we're going to draw our phospholipid bilayer or our cell membrane. All right, so there is our cell membrane. In fact, what we'll do is we'll do two layers to represent that it's that phospholipid bilayer that we've been talking about. All right, kind of not so pretty in some of these spots. Once again, this is our cell membrane. All right, so with our cell membrane, we're going to look at what we now call an organelle. So an organelle, it stands for small. One way, how would you say it? So organelle is small organs. So when you think of inside your body, you are made up of all these different organs. You have your heart, you have your lungs, you have your intestines, you have your liver, you have your kidneys. All these little things, these organs inside your cell, not inside your cell, but inside your body, help you do all the things that you do. You have your brain, you have your skin is also an organ. So your skin is kind of that outer barrier. And so in cells, we call these organelles, all right? So these organelles are what makes up these cells. And one thing about these organelles is that they're membrane bound. Membrane bound means that each one of these organelles have a membrane around them, and that membrane is that same phospholipid bilayer that we just drew and that we just talked about. And so we're going to be looking at all these different membrane bound cells that we see, not cells, but organelles that we see inside our cell. And the very first and important one is the one we were talking about earlier, is that nucleus. All right, so we talked about how the next organelle we're going to look at is what we call ribosomes. And our ribosomes, they're what we call our translators. And on our thing here, they're going to look like snowman. And later on, when we talk about genetics, we're going to see why they kind of look like these snowmen. And you know, what's a good snowman without a scarf? And actually those scarves, or these little red strips in here, these are RNA. So remember we talked how DNA is double-stranded and RNA is single-stranded. So we have these RNA single-stranded, and these RNAs have these messages. And what's going to happen is our ribosomes are going to translate. So these are our translators. they're going to translate that message from an RNA genetic code into a protein. And so that's the job of our ribosomes, is to translate RNA and create the protein that it codes for, which will then allow it to build that cell or build that organism, or however you want to phrase it. So that's why our ribosomes are called the translators. Next, we're going to be looking at the ER, or the endoplasmic reticulum. Which we just call the ER. And I'm going to draw these up here. It's going to take a second. This is a little bit more, a little bit bigger than our ribosomes. And once again, I hope you're taking out a piece of paper. If you can find some color pencils, that's always beneficial. And that you're going to end up drawing this cell here with me as well. And I suggest color pencils because you'll be able to erase as needed. So as I'm drawing our ER, there's actually two different parts to the ER. We have what we call the smooth ER, and we also have the rough ER. So in our picture here, our smooth ER is all this part here. It's the larger part of our ER. And then our rough ER is this part here, and I've got all these little dots on there. And the reason why we call it the smooth and rough ER is when scientists were first studying the cell and being able to have microscopes that could look in at the cell at a good magnification, they saw this and they saw that one aspect of it looked nice and smooth, where the other side looked really rough. And it's because of what was on these things. Now, when it comes to our ER, we've got a special name for it as well. So the special name for our ER is our manufacturing plant. All right, so we have our smooth ER, our rough ER, our ribosomes, our nucleus, our cell membrane. Now let's talk about what the endoplasmic reticulum, or our ER, actually does. So manufacturing plant probably means it's going to be making stuff, right? So, and that's what happens, is that our ER makes two different things. So within our smooth ER, it actually makes more plasma membrane, or in other words, more of that phospholipid bilayer that we talked about. And we're going to see why it needs to do that here in a minute. So our smooth ER is making a bunch of plasma membranes, bunch more membrane material needed for the cell. And then our rough ER, the reason why it's the rough ER is actually all these little dots are ribosomes. So I drew our ribosomes pretty big here, but all these little dots that are on the rough ER are also ribosomes. And because what happens is when our ribosomes are translating the genetic material, that genetic code that's found in the RNA, into a protein, it is doing that on top of the rough ER. And the reason for that is because as it creates the protein that is coded inside, or that is coded from that RNA, it then takes that protein and puts it inside of our rough ER. And then inside the rough ER, we have these special type of machines that then starts to fold the protein into whatever that protein is supposed to be. And so that happens inside here. So our rough ER helps in the manufacturing of all of our proteins. So it takes the basic protein, and sometimes you need multiple proteins that need to be put together. So there's all these little machines and stuff like that inside our rough ER that are folding and manipulating and changing and putting together our proteins for that need to be made that for the health of the cell, or for whatever they need to be done for the cell. All right, so that's the difference between smooth ER and rough ER. The other thing about the smooth ER is in here, there's a bunch of what we call cytosolic enzymes or catalytic enzymes, and these enzymes help break down toxins. And so in our smooth ER, we also have this ability to break down toxins. And we talked earlier about how inside our cells, sometimes we do things in our cells that create toxins, so we need to break those down. And so there's enzymes in here that get taken in there, they get broken down, and then they will be released back out into the environment, so it's no longer toxic to the cell. So once again, the smooth ER creates the membrane that we need for the cell. It also has enzymes that break down toxins to keep the cell healthy. And then in the rough ER is where the proteins that are created from the translator of the ribosomes are put in, and then those proteins are folded and broken down, or not broken down, but made into the product they need to be. All right, our next organelle is going to be our transport vesicles. All right, and our transport vesicles, their main job is to take proteins that were made in the ER and take them to our next organelle, which is called our Golgi apparatus. So we're going to look at these at the same time. And the Golgi apparatus, we're going to call it shipping and receiving. All right, so now we got down here our Golgi apparatus, and then we're also going to have our transport vesicles. Now our transport vesicles, they're going to stem here from our rough ER. And what's going to end up happening is they're going to create this bud. So it's going to kind of bud out from the rough ER, and inside that bud is going to be our protein. So this is the protein that was being created inside a rough yard, that manufacturing plant. And now we need to get that protein where it needs to go into the cell. And to do that, it's got to first go through the Golgi apparatus because our protein isn't quite all the way ready. And so what ends up happening is this transport vesicle transports our protein from the rough ER to the Golgi apparatus. When it gets into the Golgi apparatus, it binds to it. And the reason it can bind to it is because, like I said before, all of our organelles are membrane bound. They're membrane bound organelles, which means they have all the same type of membrane. So the membrane that's here from the rough ER is the same type of membrane that's found in the Golgi apparatus. And so we can just combine, it breaks off here, buds down, travels down here, and then it can combine with the Golgi apparatus to then allow our protein to enter into the Golgi apparatus. All because the membrane is the same. And this is why our smooth ER has to make membrane, because the membrane from the rough ER is constantly being torn off, and so the smooth ER is making more membrane for our rough ER so we can create these transport vesicles. All right, so once our protein now enters the Golgi apparatus, it has a few things that are done to it. First, it is finalized the product. So maybe in the rough ER, the protein product isn't completely finalized yet. It still needs to be changed up a little bit, or maybe another folding, or maybe another type of thing needs to be added onto it. And so in that Golgi apparatus, you have the kind of finishing up of that product, so it's ready to go. And then at the end, it is then tagged so that it can now go wherever it needs to go. And it does that as it goes through this whole space of the Golgi apparatus, and then it's going to bud out on the other end. So just like we saw it bud out here because it has that plasma membrane, it's going to bud out here because it's the same type of plasma membrane. And that protein's going to be in here. But even more importantly, there'll be one difference. And that difference is now it's a finalized product and it's got some type of tag on it. So this little thing here is going to be a tag. And that tag is going to now say, you know, this is kind of like putting a shipping label on something. So that's why we call this shipping receiving. You're receiving it here, and then you're going to ship it out. And that tag is that shipping label telling this transport vesicle where this protein needs to go in the cell, or in some parts out of the cell. And so that protein vesicle completely buds off of the Golgi apparatus, and now we'll take this protein wherever it needs to go. So let's look at a couple of examples of where this protein could be going. Let's say this is a cell inside of your mouth, and specifically one of the glands in your mouth. And it's, you know, getting close to lunchtime, you're kind of getting hungry, you're on your phone, you're surfing through your Twitter feed or your Instagram, and you see a commercial for a nice big juicy hamburger. What usually happens when you're really hungry and you see something you want to eat? So maybe it's not a hamburger, maybe whatever it is that you want to eat. Your mouth starts to water, right? And your mouth is watering because it's preparing the enzymes that are going to be needed to break down that food, because it's, your eyes are saying, oh, there's good food. Your stomach's telling you you're hungry. And so now your mouth and your glands are getting ready. So what ends up happening is you have this signal that goes from your eyes that eventually comes down to this cell here in this gland. And this cell here tells, so that signal tells the DNA, hey, guess what? We need to create some enzymes to break down food because we're about to get some food. And so we copy that DNA into an RNA form, and we're going to send that RNA outside of the cell. So here's our RNA. And it's going to go outside of the cell and find one of our ribosomes. Our ribosomes, so this RNA, and it has the code for an enzyme that helps break down food. So that RNA then finds one of these ribosomes. The ribosome reads off, translates our RNA to the protein that is supposed to be for this enzyme. And it puts that protein into the ER, so the rough ER. Then inside the rough ER, you have these different machines that are now folding it and putting it and making it the product it needs to be, creating it the exact enzyme. Then it ships that off or sends it off to our Golgi apparatus. In the Golgi apparatus, they put the final touches on it, and then they tag it, and the tag specifically says this needs to be released out into the environment. And so what ends up happening is our protein will then bind to our membrane. Our transport vesicle that had our protein will bind to our membrane. And it will combine with our plasma membrane and now send this enzyme out into the environment. And that environment would be our mouth, so that enzyme can then attach onto those food molecules and start to break them down, so that when you swallow them, they'll get into your stomach, and then they'll be broken down enough to where your stomach can start to absorb them and take those food molecules back to the cell. All right, so that's one scenario here of how we go from, oh, we need to make this enzyme, and we have the instructions for it in DNA, turn it to RNA, turn that RNA into a protein that gets built here in our manufacturing plant, the rough ER, then using a transport vesicle gets sent over to our Golgi apparatus, where it finishes the product and then tags it, and then it sends that off through another transport vesicle to go and combine with our cell membrane to release it out into the environment, and you have that enzyme to break down that food. That's that whole idea of you seeing that picture of that hamburger, you're hungry, and now this signal gets sent and you start releasing these enzymes out to allow you now to break down that food that you're about to eat. All right, now not all the time is that signal sending things outside. Like I said, sometimes it's sending it inside. So one thing I talked about is here in the rough ER, we have machines that are sitting there folding and mending and attaching different proteins together to be able to be shipped out. Well, those proteins, those machines have to be built somewhere, and they're actually encoded in our DNA here. And so you'd have a situation where, hey, we need more of these machines to build more proteins. And so what happens is you copy that code in the DNA, and it turns into an RNA. That RNA then is translated by our ribosome. It's taken here, folded up, put however it needs to be in the rough ER. Then it's shipped through our transport vesicle over to our Golgi apparatus. And the Golgi apparatus, now it is finalized and then tagged, and now it's tagged to go back to our rough ER, because now it's ready, it's completed. And so instead of now going out into the environment, because it's an enzyme that breaks down food, it's now an enzyme or a machine that builds more proteins. It will then end up here. because it's tagged. Now to go back to the ribosome, it will combine with the membrane, not the ribosome, but the rough ER. It will combine now with the membrane of the rough ER, and that protein will now enter into the rough ER, and now it's ready to help make more proteins, right? So you have this whole process, and this could be sent to other places. There's machines inside the cell, in the nucleus, I mean. So you could end up sending it to the nucleus. There's other organelles we're still going to be talking about that have machines, so we can create those machines and send them to those other organelles. So this is kind of this awesome process of going from some type of code, some type of blueprint of some type of machine, and how we actually go about creating it and sending it to wherever it needs to in the cell. All right, so now we're going to look at some other ones, but before we do that, I'm going to have to clear this up a little bit here. Give us some room to write down our other organelles. The next organelle that we are going to talk about is our lysosomes. And our lysosomes, they are our demolishing crew. So our lysosomes, what they do is they have special enzymes in them that allow them to break down larger molecules. And so what happens is, let's say the cell just got a big chunk of food, of nutrients, but it's still too big for the cell to use it the way it needs to use it. And so what will happen is the lysosomes will go find that food, connect with it, and these enzymes in here, these 4 black dots I drew, will go in and start to break down that food particle even more, or that nutrients down even more, depending on what it is. And so that's why we call it demolishing crew, because they kind of break things down. That's their main job, is to take things and break them down. Other things might happen. So we talked about how we just had to create a new enzyme here. Well, let's say some of the enzymes in here they're old and they're not working great anymore. Well, we can take those old enzymes, and in the lysosome, they'll be broken down into materials that the cell can use and recycled, and then that's when we create our new ones and brought back here. So you're keeping things fresh and new. So we're not having all these old, enzymes or machines that are building these, so that we're making sure we're having things that are doing things right and correct. There's other types of organelles out there we're going to be talking about, like the mitochondria. So mitochondria are used to create energy for the cell. Well, sometimes your cell needs a lot of energy. So if you think of like a cell in your muscle, your muscles do a lot of movement, right? And so they constantly need making lots and lots and lots of energy. Well, if you're an athlete, so let's take like the NBA for right now. So back in the beginning of March, you had all these NBA games that were happening. You had all of these athletes that were performing at their high peak. And so in their muscles, they probably had a bunch of these mitochondria because they needed lots and lots of energy. Well, what happens is coronavirus sets in. Guess what? Now now you're not needing all that energy, and so you don't need as much mitochondria, because now you have to sit at home. You're not out running, you're not lifting, you're not doing all this, you're not playing basketball like you were before. And so now in mid to end of March, you're no longer doing that, And so your body doesn't need, or your muscle cells don't need as many mitochondria. And so your lysosomes will start to attach to those mitochondria and start to break them down, because your cells want to be efficient. They don't want to be doing things that they don't need for. But let's say, all right, now they're thinking about starting the NBA next month. Well, guess what? Now those same athletes are going to be getting ready to go. They're going to be starting to work out again, run the court, doing all that stuff. And so then the cell will actually be able to create and build more mitochondria through this pathway here that we've talked about. So that's our lysosomes. All right, our next one is something we call vacuoles. Now, vacuoles are our storage containers. Now, in a lot of animal cells, especially in our human cells, we don't see vacuoles very much. But when we go back to our plant cell, so let's go back to our picture. I'm not going to bring up the one I drew, this one right here. I'm just going to draw a quick one really quick. Maybe. So here's our plant cell. There's the plasma membrane. There's the cell wall. And our plant cells are also going to have a nucleus. They're going to have ribosomes. They're going to have the ER. They're going to have the rough ER. Sorry, I'm drawing this kind of quickly. And they're also going to have the Golgi apparatus. But another thing they'll have is this vacuole. And this vacuole is the storage container for the plant cell. And specifically in the plant cell, it's going to store water, H2O. So remember we were talking earlier that plants need water to provide pressure on the cell walls so that they have structure. Well, they do this through this vacuole. This vacuole, think of it like a water balloon inside the cell. And as it fills up, it blows up, but it's not going to blow up and explode because it's going to go only as far as it can to put pressure on these cell walls. And that's what allows now our cells to be big like this. And so in plants, you'll see vacuoles in pretty much every plant cell because they need to have these water to put pressure on, have this excess amount of water to put pressure on the cell wall to allow it to have that structure. Now, in most of our animal cells, you don't see vacuoles. So I'm actually going to draw one here. I'm not going to draw one on here. But there are some animal cells that do have vacuoles, specifically in humans, and those are our adipose cells. So adipose cells are cells that hold fat, right? So as we eat more and more food, our body wants to save some of that energy that we're eating in case we get to a situation where we don't have food. And so it creates these adipose cells, these fat cells, and these fat cells have these huge vacuoles in it. And instead of having water like a plant cell would, these adipose cells will store fats and lipids and all those in there, and they kind of use that whole cell as kind of an energy bank. So if therefore I go on Survivor and now I'm not eating a lot of food for a week, it will then go and access the food in those adipose cells stored in those vacuoles. And that's why you don't see big, huge vacuoles in any other type of animal cells, because we have ways to transport food and stuff like that and nutrients to all of our other cells, and so we can just store them specifically in these adipose cells inside the animal cells. All right, so that's our vacuoles, our storage container. So they're pretty much just used to store excess foods, excess water in the source of plants just to be able to provide that energy or whatever it needs for later on down the line. All right, our next one is going to be our chloroplast. So we're actually going to keep our plant cell up here because this is one that's specific for plant cells. And our chloroplast, we're going to call it the bakery. And they're only found in plants. All right? And so our chloroplasts look kind of like these little squares. And we're going to talk a lot more about chloroplasts in chapter 7 because chloroplast is where photosynthesis takes place. And photosynthesis is how plants get energy and create energy from sunlight to be able to feed themselves. So plants don't go out and start eating. I mean, there are some carnivorous plants like the Venus flytrap and whatnot. But if you think of most plants, the only way they get nutrients and food is from the sun, the energy from the sun, and in the process of photosynthesis, which we're going to talk about. And in fact, there's so much chloroplast in plant cells. And they're green, the chloroplasts are. This is why most plants look green, is because you have so many chloroplasts in them, and that's why they see they're green. We're going to see why that is when we look in chapter 7 as well. And so really our chloroplast, once again, found only in plants here, allows for our plants to do photosynthesis to create energy from light to be able to feed the plant. All right, so then our next organelle is going to be our mitochondria. And our mitochondria is our power plant. So the power plant of the cell. This is where all the energy is made inside the cell. So these are our mitochondria. Mitochondria are also found in plants. And you might be wondering, well, if they have chloroplasts and they can make their energy, why do they need mitochondria? Well, mitochondria, what they do is they take food and they take outside energy and turn it into energy that's used for the cell. And so plants need that as well. So what happens is our chloroplasts, they make the sugars, and then they take those sugars and put them into the mitochondria, and then mitochondria creates the energy needed for the cell. Same thing happens here, but instead, animal cells, they consume food, and they take the sugars from those consumed food, give those to the mitochondria, and then the mitochondria use that sugar to create energy that is specific for the cell. And we'll talk a lot about that in chapter 6 when we look at cellular respiration. So our mitochondria are the power plant for the cell. There's all these little things happening all over here. And all these little things require energy. And it requires energy in a specific form. And that's what the mitochondria does. It takes a large form of energy, in like food or sugar, and breaks it down into a small form of energy we call ATP. We're going to talk a lot about ATP later in next chapter, chapter 5. So it takes that ATP and turns it into energy for the cell. All right, so that is our mitochondria. The next thing we're going to be looking at is something we call the cytoskeleton. All right, so we're going to erase now our plant cell. Realize that everything else we do now, these are also found in plants. We have our cell here, so we're going to erase that. We're also going to erase these guys really quick. And we're going to talk about our cytoskeleton. All right, so the cytoskeleton is something that's hard to see. It actually runs throughout the whole cell. And in fact, if we were to draw a cell, I'm not going to do it on this because we don't want to make our cell that we draw all nice and pretty look ugly. You would see that it is this set of protein beams that kind of run throughout the whole cell. And what it does is it provides structure for the cell. So as it runs throughout this whole cell, it kind of provides the structure for it. But it's very dynamic, which means it can break down one end of it and build it up on the other side really quick. And what that does is it allows this cell actually to be very fluid and allows it to move in very different directions. It can now actually allow this cell to move in a certain way or just kind of change its shape dramatically. So very dynamic. So not only is it providing the structure, so we call this the infrastructure. But it also is kind of like the roadways and bridges of the cell. So when I say, hey, we have this transport vesicle that goes from the rough ER to the Golgi apparatus, it's not just something where it's like, hey, there's this watery mess in here called the cytosol or cytoplasm, and we just send this transport vesicle off into the ocean and hope that eventually makes it here. No. What ends up happening is there's actually a part of the cytoskeleton that's running from this spot to this spot, and there's little machines. There's actually a pretty cool YouTube video. If I find it, I'll post it on the Canvas page. But there's these little machines that actually are carrying these transport vesicles from the rough ER over to the Golgi apparatus, and it's walking along this protein beam of the cytoskeleton. And so this allows for the cell to also have things in certain places. So you always find like the nucleus in a certain area and the cytoplasm in a certain area, not cytoplasm, the endoplasmic reticulum in a certain area, the Golgi apparatus, these mitochondria, they're all going to be in certain areas because of this cytoskeleton. Kind of like us humans, we have a skeleton inside of us, and in that skeleton, our organs are in certain places because they're attached to that skeleton. So if you go into like a cadaver lab, or maybe you've done this in like a high school biology lab where you open up a frog and you will find the heart's here, the lungs are here, the intestines are here, the kidneys are here, because they're all packed in there and attached to the skeleton inside of us, just like we find our organelles in certain places because they're attached to the cytoskeleton as well. And so once again, the cytoskeleton, we call that the infrastructure because it's kind of the roadways, the bridges that connect everything, and then it also holds everything into place where it needs to be at as well. All right, the last thing that we're going to be talking about on our cell is how the cell, how some cells have certain appendages on them that allow them to be able to move. And those appendages are called cilia and flagella. So our cilia and flagella, these are, you kind of want to say like the oars or the motor. How would I say this? Let me look how I have it written down. So the oars and propellers of a cell. I wanted to say motor, but that's not what I wanted. Propellers of the cell. And so when we're looking at cilia are these small kind of hair-like things that come out of the cell, but they're not hairs. They're actually part of the cell. They're embedded into the membrane of the cell, and they're all over the cell. And this allows the cell to do different things. And so one thing to realize, not every cell has cilia, but those that do, they will use this for two things. Either to have the cell move, so they'll row, so think of like a Viking boat where you have all these people rowing in the same direction, and they'll row and allow the cell to move forward that way. So if this is a single cellular organism that has cilia, this allows them to move back and forth and move around in their environment by these cilia acting as rowboats. The other thing these cilia can do is allow them to attach onto things. So maybe attach to another cell, or maybe attach to some type of thing in the environment. So these cilia allow them for movement, also to attach to areas in the environment. Now flagella are much bigger. And a classic cell that has flagella is the sperm cell. So a lot of you have seen like a sperm cell. It kind of looks like a tadpole. And this is your flagella right here. All right, so this is more like a propeller, and it will kind of spin around like a propeller to allow the cell to move forward. Now, flagella are only going to be on one polar end, so it's either going to be on this end or that end, because that will then propel the cell to go the opposite direction where you see the propeller. So the propeller's on this side, then it's going to go in that opposite direction. It's going to go that way. And cells can have anywhere between 1 to 3 flagella. So here's one with 1. You could do one like this with 2. And then you also have some that would have 3. But we don't have any more that have more than 3. And once again, they use these flagella to allow them to move forward. And most cells don't have flagella, but like I said, we have sperm cells that have flagella. There's other single cellular organisms that have flagella that are eukaryotes. And so we just kind of wanted to talk about those as well. All right, so this is the end of chapter 4, the tour of the cell. We got to see a lot of different things about the cell. We got to see all the little different things that are going on inside the cell. And what we're going to end up seeing in these next chapters, chapters 5, chapter 6, and chapter 7, is actually some of this stuff, how it works, right? So in chapter 5, we're going to talk about energy and how that energy is conserved. And we're going to look at, all right, how does the mitochondria create energy? How does it create that ATP? And then if we're going back to our plant cell, how does the chloroplast take sunlight and turn it into sugars to create energy as well? And then after that, so that's chapters 5, 6, and 7. And then we'll do chapters 8 and 9, and that's where we'll learn more about genetics. And we'll dig deeper into the nucleus and how do we actually go from a DNA to an RNA to a ribosome to a protein, and how does that whole process work as well. So actually dig deeper into that. And so while we're just kind of doing a tour of the cell now, we're going to take a stop at some of these tour sites and learn a little bit more about those in the coming chapters.